Well, welcome back to our next session here in our September NBAC meeting. Our next panel is called Science Review, New Research to Inform Future HIV Vaccine Development. As the HIV epidemic enters its fifth decade, scientists are still pouring in their time and resources into developing vaccine candidates for this virus. In this session, we'll hear from five speakers who will provide an overview of new research to advance scientific knowledge on stimulating the rare antibodies needed to prevent HIV. Our speakers include Dr. James Kublin from the HIV Vaccine Trials Network, Dr. William Schief, who works at IAVI, Moderna, and the Scripps Research Institute, Dr. Facunda Batista from both the Ragon Institute and Massachusetts General Hospital and the Massachusetts Institute of Technology, Dr. John Steichen from the Scripps Research Institute, and Dr. Torben Schiffner from the Scripps Research Institute. Dr. Kublin?
Thank you very much. And I'll be sharing my slides.
Right. I see your slides are up. You have the floor, sir.
Thank you very much. So, and thank you to the organizers for the invitation to speak today, and to HHS as well for the tremendous support of training and education, surveillance and epi, and the R&D for vaccines that benefit all Americans and indeed the world. And apologies for not being with you in person. So I'm a physician scientist. I was working as a phlebotomist in the early 80s when HIV was initially identified. And in these past 40 years, we have struggled and made strides toward HIV, an HIV vaccine. I'll provide a brief introduction so my colleagues can explain in further detail why we are optimistic that the progress we're seeing is uniting us in unprecedented ways. But first, a brief snapshot on HIV epidemiology and where we are today. It remains a major global public health issue, having claimed over 40 million lives to date, and transmission is ongoing in all countries globally. There was an estimated 39.9 million people living with HIV at the end of 2023, 65% of whom are in the WHO African region. And last year, an estimated 630,000 people died from HIV-related causes, and an estimated 1.3 million people acquired HIV. And as you can see on the figures on the right-hand side, the number of new infections has indeed come down, but nowhere near the target for UNAIDS will be met in 2030. So the cost to society due to HIV is really quite dramatic. The global drug market size is valued at over $30 billion a couple of years ago and is poised to grow. And the US government invests in the domestic response to HIV with over $28 billion per year. That is the cost to society. And the average lifetime HIV-related medical cost for a person with HIV in the US is estimated to be over $420,000 discounted, over $1 million undiscounted. And these costs far exceed that of any other preventive communicable disease, and indeed are multifold over other non-communicable diseases such as diabetes and heart disease. So we at the HIV Vaccine Trials Network focus on our mission daily, which is to fully characterize the safety, immunogenicity, and efficacy of HIV vaccine candidates with the goal of developing a safe and effective vaccine as rapidly as possible for prevention of HIV infection globally. And we do this by reaching out to global colleagues, and collaboration in this effort, of course, is essential. We apply social behavioral research due to the tremendous vulnerability of some of the populations who are exposed to HIV, and work across the fundamental and basic immunology and vaccinology to apply leading technology to advance our mission. I also noted that earlier or yesterday in your agenda, you were focused on TB vaccines. It was about 7 years ago, Dr. Fauci asked us to also take on and assisting with the development of TB vaccines. So we are the world's largest publicly funded clinical trials program, which is why 4.5 years ago, we were also asked to lend our expertise and leverage the tremendous global footprint of over 100 sites to the COVID vaccine development effort. We are funded in large part by the Division of AIDS at the National Institute of Allergy and Infectious Diseases at NIH, with additional projects and support by a number of foundations and biotech pharma when we can engage them. We also have a very focused discovery medicine program, and this is a series of studies that are focused on developing as rapidly as clinical trial rules allow an iterative process of testing early candidate immunogens to induce germline binding, for example, and you'll hear more about this specifically from Bill Sheaf and colleagues from Scripps. So an example of that, and I'm highlighting here on the left, the spike protein of HIV, or envelope, that has been interrogated incredibly over the past 40 years as to why it's so difficult to induce binding antibodies and particularly neutralizing antibodies that are effective in preventing HIV acquisition. We have identified these 5 envelope epitope sites that are particular targets for HIV neutralization and have a number of priming immunogen types that are in the pipeline of the HVTN to focus the immune response to the induction of these broadly neutralizing antibodies, or BNABs. But there are many challenges, and this was highlighted over 15 years ago by one of our principal investigators, Dan Baruch, in the development of a prophylactic HIV vaccine. Most of these challenges remain. The viral clade and diversity is something I'll speak to a little bit more, as well as the early establishment of latent reservoirs. Being a retrovirus, these reservoirs persist for life. Their immune correlates of protection are unclear. Viral evasion of both humoral and cellular immune responses is a very distinguishing characteristic of HIV. And these antibody responses that are elicited are typically type-specific. No method exists currently to elicit broadly reactive neutralizing antibodies in humans, although this is particularly an area in which progress over the past decade will be presented to you by my colleagues in a moment. And other reasons such as no confirmed early small animal model that is directly applicable to humans. And as the challenges in biological plausibility of an HIV vaccine have not been yet confirmed, there's little pharmaceutical interest. I mentioned the diversity of the trimer or envelope spike protein, which is highlighted on the left in a cartoon illustrating the diversity of the global influenza 1996 pandemic on the upper left, and individuals and their respective diversity illustrated in the cartoon, as well as the Amsterdam cohort and Congo in 1997, trying to illustrate that diversity in comparison to the diversity of HIV today would indeed fill the entire screen. So HIV envelope is quite distinct in both the sequence variability that you see on the bottom in purple, as well as the sugar moieties of glycosylation that occurs on the surface of HIV envelope. And you can see how densely these spike proteins or envelope is populated by both green and purple in comparison to the influenza virus or the RSV fusion protein, which is more similar to SARS-CoV-2 coronavirus. Another unique characteristic of HIV is the lack of density of the spike proteins on the surface. In the top, you see electron micrographs of influenza and measles with this corona-like density of the spike protein on the surface. And in the bottom left, a cartoon of SARS-CoV-2, and that of HIV in blue on the bottom left in the spherical cartoon with these low-density white spike proteins. And this does not, therefore, permit the interspike cross-linking that is oftentimes a characteristic of neutralizing antibodies against known, well-known, and well-controlled global potential pandemic viruses. These illustrate just a couple of the challenges, and this cartoon of how late broadly neutralizing antibodies are induced in an individual who's acquired HIV is illustrated here. The initial viremia occurs very early in infection. That is controlled, fortunately, in most people by their immune response, but not sufficient to eliminate the virus. Seroconversion occurs. These autologous neutralizing antibodies may occur, but broadly reactive neutralizing antibodies, or BNAbs, only occur in a minority of individuals who have acquired HIV, and it's these BNAbs that are our holy grail or goal in our discovery medicine program, and indeed much of the work supported by HHS through NIAID's CHOV-D programs, such as at Scripps, that you'll hear about. HIV also has this multidimensional nature of its reservoir. As I mentioned earlier, being a retrovirus, it integrates into the genome in resting cells that are dormant in many different tissues around the body, that is illustrated here. Now, many people have looked at the impact of an HIV vaccine, and that's the impact of not even a highly efficacious vaccine, such as we've seen with SARS-CoV-2, but a partially effective vaccine of only 50%, having a really very dramatic impact on HIV as modeled on the left, and the potential cost effectiveness of pre-exposure prophylaxis, or PrEP, independent or combined with an HIV vaccine, is illustrated in the cartoon on the right. This is a publication that we worked on over 5 years ago, illustrating how indeed cost-effective an HIV vaccine would be, and particularly in combination with PrEP. And as you've heard, progress over the past year has demonstrated much accomplishments in the administrations of PrEP. But when these initial PrEP findings were revealed 10 years ago, Drs. Fauci and Marston published a perspective in the New England Journal of Medicine stating, ultimately, we believe the only guarantee of a sustained end of the AIDS pandemic is in a combination of both non-vaccine prevention methods and the development of a safe and effective HIV vaccine. 10 years later, this summer at the International AIDS Society meeting, now the current director of NIAID, Jeannie Marazzo, stated, we at NIAID, NIH, and I think in general, the scientific field are 100% committed to continuing to work full speed and very deeply on the development of an HIV vaccine. And this is the landscape of HIV prevention timelines late last year. For the future in 2024, 25, 26, there is currently no large efficacy trial of an HIV vaccine, and our focus is to change that with the development of antibodies and active vaccination strategies that can elicit, through active vaccination, these broadly neutralizing antibodies. And again, you'll hear in great detail from my colleagues about this momentarily. I wanted to highlight just briefly the tremendous leveraging and collateral benefits, so to speak, of HIV vaccine research and development that has put into the field over the past 2 decades, progress that resulted in COVID vaccine development. Notably, last year, the Nobel Prize awarded to Drs. Weissman and Carrico on the advances that they made for the possibility of these mRNA platforms was indeed initially funded and supported through NIAID for HIV vaccine development to Drew Weissman at UPenn and others. And indeed, many of the monoclonal antibody discoveries that have occurred in HIV have benefited immuno-oncology, the vectors that have been explored and tested for HIV vaccines, such as adenovirus, DNA plasmids, and a number of other technologies have been applied to advance not only COVID vaccines, but as I mentioned, other immunotherapeutic and prevention modalities. So we think that the response to the SARS-CoV-2 pandemic has shown what a global singular focus can achieve. And this attention to these pandemics must be a lasting moral obligation. And we can apply this momentum, enthusiasm, and resources that COVID-19 vaccines have brought to the world to make the possibility of achieving an HIV vaccine a reality. And last year, in a special entire journal article and an entire issue from Current Opinions on HIV/AIDS, I provided indeed the answer to whether or not an HIV vaccine is achievable, and it is in large part a result of the work that we're doing at the HVTN and that our colleagues are doing at Scripps, and that through this Discovery Medicine program will make the advances necessary in the coming years to make HIV impossible to ignore. I mean, for the past 40 years, we've lived in the presence of this virus. It was once our greatest pandemic concern with heightened awareness. It has now taken a backseat to other social and health issues facing us. And we are now challenged with bringing it to the forefront again so that ultimately we can control this global pandemic. So with that, I'd like to thank you all for your attention and turn it over to my colleagues at Scripps.
Thank you very much. Our next presenter is Dr. William Schief.
Hello, and thank you very much for having us. Let me get the laser pointer here. All right. As Jim said, we and all of our collaborators have a lot of optimism about, and we still feel a lot of importance of trying to develop a vaccine. We have a lot of optimism about the fact that we think we can make an HIV vaccine a safe and effective one. And I think the rest of us, me and Facundo and John and Torben, will try to convey what progress we've made and where are we trying to go. Just to echo what Jim said, he gave a lot of nice intro, and I just wanted to highlight, we do believe this is it remains a super important problem. Last year, there were 40 million people living with HIV, but 23% of those were not receiving antiretroviral treatment. That's more than 9 million people who are, at risk and are transmitting. The Global Fund for AIDS Response is around $20 billion in constant 2016 dollars. It's less than what's needed by something like $10 billion and is going down rather than going up. It's paying for a lot of drugs for a lot of people. And long-acting PrEP can, as we've learned, can be highly effective. And we believe it should be maximally deployed. But, it faces many challenges to global access and long-term use. And so, as Jim said, we really do need a vaccine. It's a super important aspect of global health. So Jim showed some images of the spike. This is a video of the spike with glycans. The blue structures are highly flexible glycans on the outside. And those are very hard for antibodies to bind to. As you know from the COVID vaccine, the primary aim for the development of an HIV vaccine is to, I don't know why it stopped, sorry, is to, actually, I'll just leave it. The primary aim is to elicit neutralizing antibodies that'll block the spike from interacting with CD4 and CCR5, the receptors on the receptor and co-receptor on human cells. And on this trimeric spike, the red portions are highly variable. They change very much from one strain to another. And the portions that we colored yellow are less than, they're not super highly conserved. So if you're an antibody and you're trying to be a broadly neutralizing antibody that can neutralize diverse HIV isolates, you definitely cannot touch the red portions, and you got to negotiate with the yellow. So it's very difficult to find a place to land on the HIV envelope to be a broadly neutralizing antibody. So the structure, glycosylation, and sequence variation pose major challenges to the development of broadly neutralizing antibodies. But through the work of many in the field, including Dennis Burton at Scripps and John Mascola at the NIH Vaccine Research Center, Michelle Nussenzweig, and many others, many highly potent broadly neutralizing antibodies against HIV have been discovered. They do neutralize diverse isolates. This is a cartoon of a trimer with 4 different classes of broadly neutralizing antibodies bound. They can protect against infection in non-human primates and humans. Different classes bind to different epitopes on the HIV envelope, and we believe an HIV vaccine will need to induce multiple classes of BNAbs in order to achieve sufficient neutralization, potency, and breadth to protect against the diverse isolates circulating among humans. And so in my lab and with all of our collaborators, including Facundo, who's going to speak next, and Shane Crotty at LJI, Dennis Burton at Scripps, many others, we have very strong and highly developed programs to develop vaccine components that induce broadly neutralizing antibodies against each of these 4 different parts of the spike. And we've published in the last couple of years many papers this year. So you can read, I do not have time in this short talk to review all of our progress, but we're making a lot of progress with our collaborators, and we're very excited about progress in this direction. So how are we approaching this, and what's the strategy? So that what we need to think about is, well, what do we need to achieve? The way we think about it, you want to start from human naive B cells, and the vaccine needs to trigger the right B cells. Here's a red one, would be one of the correct ones that we want to trigger. This red B cell has the genetic components in its antibodies that give it the potential to develop broadly neutralizing antibodies. And these blue ones have other antibodies on their surface that don't have the genetic potential to develop into BNAb. So the vaccine needs to first trigger the right naive B cells, and then it needs to expand them and drive them to mature and gain somatic hypermutation in their B cell receptors, and finally convert into plasma cells and secrete broadly neutralizing antibodies. And that sounds like a very linear and straightforward process, but it's actually extremely difficult. First of all, the right precursors they're very rare. On average, something about 1 in a million, but some are much less common than that. I mean, your vaccine needs to find them and turn them on. They have specific genetic features that differ from one BNAb to another. So your vaccine needs to know how to go find the right ones with the right genetic features for that particular class of BNAb, turn those on and get them to start maturing in the right direction. And for each class of BNAb, it's not just one precursor, it's a diverse pool that all share the right genetic features that give a potential to develop that kind of BNAb, but are otherwise diverse. So you have to hit with high specificity, but you also need to have this sort of breadth of triggering of the right precursor pool. And as we know from lots of research going back about 20 years, the precursor B cells are really hard to activate with wild type HIV proteins. So we have to redesign the HIV proteins to get them to turn on these B cells. So just starting the process is really difficult. And then we need to select for a lot of somatic hypermutation to give the antibodies the ability to bind really tightly to specific sites on the trimer that don't vary too much. It's a really hard structural biology problem. And so the strategy that we're pursuing is called germline targeting vaccine design, and it's really anchored off that priming step where you design an immunogen that can find the right naive B cells and turn them on, get them to mature a little bit, gain a little bit of somatic hypermutation, and produce a pool of germinal center and memory B cells that can then be triggered with a subsequent immunogen that's closer in structure to the native HIV trimer, and kick the system further in the right direction, and ultimately through a sequence of different immunogens, starting from a germline targeting priming immunogen and ending up, we believe, in native HIV trimers like the one I showed you in the movie. We think we can carry out this process and produce plasma cells that secrete BNAbs. This is a very complex vaccine strategy that, hasn't been used in any human licensed vaccine. But it does have the advantage that at every stage, at every step, you know what you're looking for. So when you do a vaccine clinical trial, you can prove to yourself that you did or you did not trigger the right B cells and generate the pool that you were the pool that you were looking for. And then on the next one, you can tell, did we advance the maturation further? So it really allows for engineering optimization, and you can answer yes or no. And if the answer is no, you can go back to the drawing board and figure out what'd you do wrong. It's not a black box. And so we really like that about this vaccine strategy. Now, if you're going to make a vaccine it's going to really help people, and you're going to immunize a lot of people, you obviously need everybody that is vaccinated to produce broadly neutralizing antibodies, or at least a very high fraction of them. And we believe right from the start, you need to make sure that the priming vaccination works. So over the last 15 years, we've placed a lot of emphasis on making sure we get the right germline targeting prime immunogens developed and delivered. And Facundo and Torben and John will talk to you about data from those. I'm just going to tell you about one example from a human clinical trial, and I'm not going to say more on this page. So the one example I'm going to talk about is the VRC01 class of BNAbs. We have lots of structures of them, and there's many examples of them. They bind to the CD4 binding site, and the heavy chain of the VRC01 class antibody structurally mimics CD4 in the way CD4 binds to the HIV trimer, and then the light chain is sort of extra baggage that has to squeeze into the CD4 binding site. And the way these antibodies do it is they use specific heavy chains using specific VH1/2 human V genes with specific alleles of those V genes to mimic CD4. And then to allow the light chain to squeeze in there and not bump in to the trimer, they just need a very short 5 amino acid light chain CDR3 to avoid the clash. Otherwise, they're very diverse. These antibodies can have very different heavy chain CDR3s, which is something that's normally very specific for an antibody interaction. In this particular case, it's unusual. They can use diverse heavy chain CDR3s, and they can use diverse light chains. And so we need a priming immunogen with appreciable affinity and avidity for diverse VRC01 class human naive precursors if we're going to have a chance to start the process of making those BNAbs. And so over the last many years, we developed this nanoparticle immunogen called the EOD-GT860mer. It's a self-assembling nanoparticle presenting 60 copies of an engineered GP120 outer domain. It has appreciable affinity and avidity for diverse VRC01 class human naive precursors. It primes VRC01 class responses in stringent mouse models, and it induces VRC01 class memory responses that can be boosted toward BNAb development in mouse models. And working with many different collaborators, including Facundo, who's talking to me next, we've had the pleasure of working on lots of different mouse models. And Jim mentioned that one of the concerns about HIV is we don't have any good small animal models, which is true for HIV infection. But actually, over the last several years, working with Facundo and others, David Namazi, Fred Alt at Harvard, the field has shown that mice can be incredibly useful, and they have really accelerated progress toward HIV vaccine development. And I will say, in our clinical data, we see echoes of the mouse results, and it's been quite predictive and super helpful. So the one piece of data I want to show you from a human clinical trial is this paper we published in December 2022. It was a test of this immunogen EOD-GT860mer in humans, and the goal was to immunize humans and ask, does that vaccination produce VRC01 class precursors? Did it induce that response? And it did do that in 97% of the participants. And the one participant that didn't make a VRC01 class response actually was genetically incapable. Their VH1/2 alleles were not the right alleles. And a small percent of humans have that issue for this class, which is one of the reasons why we want to induce multiple different classes of broadly neutralizing antibodies. But we think this paper was really important because it demonstrated for the first time that this rather complicated strategy, it showed that at least the first step really can work in humans where we need it to work. And it told the field, hey, let's everyone start working on this because we believe it has legs. And so that trial established proof of principle for germline targeting vaccine priming in humans. The next major milestones are, if you're going to induce BNAbs, you can't just have the first step work. You need to have these successive steps work. And the major concept there is heterologous boost. We need to show that we can give a different immunogen and we can actually advance the maturation of the B cells in the right direction. And that's being tested in IAVI G002 clinical trial. It's actually a manuscript that we've submitted for publication, and it was carried out in collaboration with Moderna. The vaccines were delivered as RNA to help accelerate the process. And if we can show that that works, we need to start marching forward and try to actually induce broadly neutralizing antibodies in humans for one class. And then finally, to make a vaccine, or at least to get something that we think would merit phase 2 human clinical testing, we need to be able to induce multiple BNAb classes in parallel. And I have this line at the bottom is that in addition, we think it's really important to establish these milestones, certainly in the United States, but also in particular in Africa, where the vaccine is most needed. And in this clinical trial, IAVI G003, we tested the same priming immunogen as in G001. We tested it in Rwanda and South Africa, and we're preparing that publication for submission. And I just mentioned that we're working with Moderna to accelerate vaccine development. And it's particularly important because we're going to have to do multiple phase 1 clinical trials to learn how to develop this rather complicated sequential regimen. It's going to require producing multiple clinical immunogens and carry out multiple clinical trials, and RNA is very enabling there. RNA also allows the delivery of membrane-bound trimers, which we, with Facundo and others, have shown actually have a lot of benefit for vaccine responses. And so, as Jim mentioned, all of this requires tons of collaboration, lots of people thinking and talking and working together. And obviously we have that. Our work is anchored with our Scripps CHOV-D grant funded by NIAID and our CAVD grant from the Gates Foundation, the IAVI Neutralizing Antibody Consortium. We work with Moderna. Our work in Africa has a lot of sponsorship from PEPFAR and USAID, and we obviously have tons of collaboration. So I'll just want to, and I just want to acknowledge the study volunteers for G001 and all the many, many people that contributed to that clinical data. And I'll stop there and say thank you.
Thank you very much. Our next presenter is Dr. Facundo Batista.
Yes, thank you very much for the opportunity to present here. It is an honor to present in front of this committee, and I apologize not to be there today, and I'm just presenting here from Boston. So I will continue with the theme of Jim and Bill, actually, and I will give you my perspective. I am a basic B-cell biologist that has worked all my career around B-cells, and I will kind of reiterate a little the message of Bill in more the context of the B-cell biology. And as Bill mentioned, antibodies are initially expressed as B-cell receptors on the surface of B-cells, and the genes encoding for the variable domains that are responsible to binding to the antigen are in naive conformation without any mutation. So upon vaccination or upon viral infection, those B-cells will go into a particular organ, the lymph node, and inside that organ will form these small entities that are called germinal centers. And here is where the B-cells are going to do something magic that is the only B-cell in the whole organism that do this, that is, they will start to accumulate somatic mutations in the variable regions of the genes. And as a result of that, initially these antibodies that are very low affinity, then they will become very high affinity, and they are going to be able to differentiate into plasma cells that they are going to produce these antibodies from our bone marrow, or into memory cells that they are going to activate by the same boosting or same viral infection later in life, producing this cycle again. Traditionally, vaccines has been empiric, and the empiric vaccines deal particularly to the initiation of this immunoresponse. And for a classical response to flu or COVID-19, you need to accumulate 3 or 4 mutations. So this is a very short cycle of the GC. One particularity of these broadly neutralizing antibodies that Bill nicely introduced is that the number of mutation and the level of maturation. These antibodies require, in many cases, between 12 to 25 different mutations to accumulate. And this cannot be reached just with one shot of vaccination. So what Bill mentioned in the previous slide is recapitulated here, and I have not been part originally of this, but I joined with a lot of excitement, is the notion that we can guide B cells through different shots of vaccines through the process of starting to accumulate somatic mutations in a way that they will then differentiate into broadly neutralizing antibodies. And I think that in the context of vaccine, again, this is unprecedented. Traditionally, people will work on the basis of an inactivated virus or bacteria or a single shot of vaccine that will lead you into these antibodies that are going to be very good at neutralizing flu, malaria, COVID-19. However, in the case of the HIV, this poses an unprecedented scientific challenge that is the one of how we can guide those B cells toward the differentiation and how we can guide them through acquiring the right mutations in the process. So the initial step of this vaccination, and this is the same paper that Bill mentioned that is part of his work and his many collaborations, in my view, is unprecedented. And it's the demonstration that we can trigger the right precursors in humans by these molecules, that is the GTA60mer. And why it's unprecedented is because the response in this particular case has been followed in an unprecedented way and has opened the way to prove indeed that all these assays that were initially characterized in mice, they can be directly translated into humans. In other words, vaccine predictions that can be done in the mouse, and this was not done only by us, but previous to us by many, many groups that are cited here, they can then be mimicked in the response they have put into humans. So what was the challenge here? I mean, the challenge is that these mice are humanized. And what I mean by humanized is that we transform the response of the mice, instead of carrying antibodies from the mouse, to carry antibodies in which the binding site of the antibodies are now replaced by humanized B-cell receptors. And one really big innovative issue that was in the field is that producing these mice traditionally will take several years. And what we have done here at the Reagan Institute in Boston is that we develop a technology that allow us, by genetically modifying direct eggs of the mice, that allow us to establish these humanized mouse models in just a matter of months. So this has the potential really to kind of translate and generate many of these antibody variants that allow us to evaluate the many immunogens that are required to evaluate, to then being able to iterate in fast and cheap animal models to them for applications into humans. And one point that Jim made that I think that is fundamental is how the discoveries that we do around the HIV vaccine can be translated into other fields. And we have shown and others that this technology that we established of very quickly generated is now having an impact not only on HIV, but it's having an impact in flu, malaria, COVID-19, and other diseases. So what is our experimental setting? And essentially what we can do then is we can make this mouse in which we have humanized the B-cell receptor, then we can adopt it transfer. And what we do this is because we want to recreate the physiology or this rare precursor frequency that Bill has established. And we work at rare precursor frequencies that mimicking what it has been offered in humans. And this is where we can test large cohort of these animals in response to protein, in response to RNA, in response to different formulation of proteins, in response to different nanoparticles, follow the germinal centers that are these particular structures that form among the immunization, and then follow many aspects of this immune response. And I will go into details of this, but I would like to highlight a couple of important innovative technologies. One is this one that was developed again in collaboration, but is essentially the work of Andrew Warsa's script, and this has Andrew is an expert in cryo-EM. And what Andrew has shown, I mean, based on the collaboration that we have with Bill and our lab, is that he can directly read the antibodies that are produced. This is the trimer of the protein, and he can incubate the trimer with a sera that we develop on those antibodies. And by using this incredibly innovative technology that he has developed in his lab, he can really show that the antibodies that are coming from the humanized B cells are able to go deep into the pocket that is required to become a broadly neutralizing antibodies. What is important too is that he's able also to show that antibodies that are developed by the blue cells from the mouse, not the humanized, they are binding to other parts of the proteins, and these antibodies will never become broadly neutralizing antibodies. So this kind of resolution or this kind of technology shows the impact that this has in the collaboration has on this, but also how much we can learn by working together and bring my expertise on visual biology with Bill expertise in immunogen design, with other people expertise in cryo-EM, with people working on different adjuvants that allow us to make this response more potent, etc., etc. So we can follow the evolution of the germline cells by counting the mutations that these cells are able to acquire in the germinal centers. We can follow that these cells are able to diversify. And this is critically important. This diversification is critically important because it is at the basis of then going to the next stage and generating the variants that are going to bind to the next guiding immunogen. We can also are able to express those antibodies and show that they increase the affinity for the original antigen. And here I will go very quickly to recapitulate, 3 papers that again has been the result of an incredible collaboration between our lab and Bill and many others, actually. And the first one is the one that we show very recently published in Science. And again, this focus in this particular D3 glycan or N332 binding site. And essentially, just to summarize this in one slide, what we have shown is that in these mouse models that we create, we can really preclinically validate this immunogen N332GT5 that has been produced by John Stein in the laboratory of Bill initially as a good candidate for priming immunogen that can eventually go to the clinic. What we have also shown is that we have not only used this as a primary immunogen, but we have characterized 2 potential boosting immunogens that are able to shepherd the response into the right direction. And we have evaluated in this study 2 methods of delivery. One is protein, and we have compared also with RNA. And we have shown, or we have evidence that the membrane configuration of RNA seems to be more potent at actually priming the response to guide this into the germinal centers, and then being able to boost this response to kind of continue to shepherd the cells in the right way. In a second study, and now not focusing the N332 binding side, but then focusing the CD4 binding side, and Bill already referred to this because again, it's a collaboration that was done from my lab, his lab, and many, many others. What we have shown is that this fast generation of humanized mouse models in native human BCRs can really inform this vaccine design very, very quickly. We have indeed show a novel preclinical validation of GT8 60mer and GT8 60mer protein, and we have also provided evidence for the right in boosting or preclinical validation of the boosting that not only prime the right precursors at very rare frequencies, but they are also to guide those precursors into the right path that guide them towards becoming a broadly neutralizing antibody. So this work, together with the work that published together with Chris in the laboratory of Field, constitute the preclinical validation for this G002 that Bill has mentioned that is hopefully going to publish very, very soon by his lab. So we have published a 3rd study, and this again focusing the work in a collaboration with again with Bill and together with Torben in his lab that focus in MPER. And very, very similarly in this study, we have shown the preclinical validation. We have contributed in part to the preclinical validation. And here we have also learned very interesting biology. We have learned the important biology that how cells that are undesired competitors for this, they initially play a role, but we have solved this problem by creating immunogens, by Torben creating immunogens that has much higher affinity for this. And in that way, actually, those immunogens are able to now guide cells into the GC and keep them there for a very prolonged period of time. So keeping up on the time, I want to kind of leave you with 2 particular messages. I think that the work that we have been doing in collaboration with Phil, our expertise in visual biology and mouse models with expertise in immunogen design has led to an incredible number of publications. All of them have enabled clinical trials, and that now these immunogens are being tested, not in the mouse, but now really in humans, and in particular this N332GT5 and this Moderna GT002 Moderna clinical trial. And just to finish, I mean, the work that we have been carrying until now has particularly focused in the prime of the right cells, and we demonstrated that that can be done in a very low and rare precursor frequencies of the B cells. We have also carried this work of starting to explore the boosting, and we can see that we are starting to guide those cells into the right direction. But of course, there is a long way to go in the developing of broadly neutralizing antibodies, and that is something that we are putting a lot of efforts right now, and is something that leave us with plenty of work to do. And in that context, what we are doing indeed in the lab, we are not developing only humanized mouse models or non-human primates mouse models, but we are also working particularly in the late state of the immunoresponse by generating mouse models that now reflect later parts of the immunoresponse to be able to concomitant and validate not only immunogens in the early stage, but also in the late stage of the immunoresponse. And with that, I would like to finish by acknowledging a ton of collaborations, but in particular, we have a very long-standing and very robust collaboration with the laboratory of Bill Schiff and many of his members, John that will be presenting next, and Torvin who will be presenting next, and the laboratory of Andrew Wirtz and many others. And our source of funding, again, what has nucleated us with all this different expertise was this CHAV ID, the CIVD products provided by the Melinda Gates Foundation, and several grants that we received through the National Institute of Health. And with that, I would like to really thank you. It's an honor to be presenting to you today. And I will pass to the next speaker, that is John's statement, that is based at the scripts.
Thank you very much. Our next speaker is Dr. John Steichen.
All right. Hello, everybody. Thanks for inviting me. So I'm going to be talking about vaccine priming of rare HIV broadly neutralizing antibody precursors. And so the main focus of this talk will be on these two papers that we published in Science. One was in 2019 and one in this year. And the main focus is on this strategy called HCDR3 dependent germline targeting. This is sort of a extension of what Bill had talked about and Facundo. So in the case that Bill talked about, that was sort of a special case that a lot of labs pursued initially. It's an antibody called VRC01, and it's a special case because it's not dependent on its HCDR3 for binding, which is unusual for an antibody. And so to extend that, we needed, because we know we need to target many bNAbs, and so we needed a strategy to target specifically HCDR3 dependent antibodies. And so this first paper in 2019, we described an immunogen design strategy for targeting HCDR3 dependent antibodies. And then our most recent paper, we described the validation of the method in rhesus macaques. And I'm going to mainly focus on this validation study from this year. And so the antibody we're targeting is called BG18. This is a HIV broadly neutralizing antibody that binds to the V3 glycan epitope. It's called a glycan epitope because it's basically surrounded by sugar molecules. So this structure on the right is showing the HIV envelope protein in gray that's on the surface of the virus. It's covered in these sugar molecules shown in green. I'm showing a few of them here. And that basically masks the virus from the immune system. And BG18 has this blue loop shown here that can reach past the sugar molecules. And that's called the HCDR3 loop. So it uses that HCDR3 loop to reach through the glycans, and that allows it to contact the conserved surface of the virus underneath. And so this antibody can neutralize about 63% of global HIV isolates. We believe the precursors that can develop into BG18 are present in most people. So we think most people should be able to make a BG18 class response. So we think it's a good vaccine target. And the one challenge of this is that the precursors occur at a very low frequency. So they're much lower than VRC01, which is in part due to the dependence on the HCDR3 for binding. And this is essentially the lowest frequency precursor that we've demonstrated successful priming of. And as I said, the HCDR3 binding mode is the normal way that antibodies bind. So if we can make this strategy work, then we think we can make a vaccine using germline targeting for essentially any pathogen. And so as a proof of principle, we started targeting this antibody BG18. We developed an immunogen called N332GT5, which Facundo has just talked about briefly, using this HCDR3 dependent germline targeting strategy. And the way we define a BG18 class antibody is through a sequence motif in the CDRH3. So in this blue loop, if you sequence the antibody gene in a B cell, you can look at the HCDR3 sequence and see if it has this sequence motif, this amino acid sequence, then you classify it as BG18. And so, as other people have touched on, one of the challenges for germline targeting is that you're specifically targeting human genes, human antibody genes. And so other animal models don't have exactly the human genes. And so most animals, it's too different to actually use. So that's why Facundo has been so valuable, because he can knock in the human genes into mice. But in the case of rhesus macaques, they sometimes do have antibody genes that are similar enough that you can target them. And so here we looked in rhesus macaques and their naive B cells, and we found that when we looked at 60 rhesus macaques, we found the majority of them do have BG18 class antibodies, and the frequency is just a little bit lower than it is in humans. It's about 8-fold lower in rhesus macaques compared to humans. So we think this animal model is challenging, but it's somewhat realistic. You know, it's pretty close to humans. And so we carried out an experiment in macaques, 8 macaques. We did with N332GT5 as the immunogen. We did an escalating dose prime, meaning that you give an exponentially increasing amount of immunogen over about 2 weeks. This strategy was developed by Shane Crotty and Daryl Irvine. They just found that it gives a very robust immune response. And then we gave a bolus boost at week 10. The adjuvant is SMNP, which is also a very strong adjuvant developed by Daryl Irvine. And the primary readout is we're looking for BG18 class B cells or antibodies. And we use B cell sorting and sequencing of the antibody gene or the B cell receptor. And so the B cell sorting and sequencing was done by Shane Crotty and Devin Stock's labs in this case. And so what we found was when we looked in lymph nodes after immunization at the germinal center B cells, we found BG18 class B cells in 7 of 8 macaques. In contrast to it, we had a control group of animals that received a wild type HIV trimer, and we did not detect any BG18 class B cells in those animals. So we think the germline targeting strategy was necessary. We also looked in the blood at memory B cells, and again, we found BG18 class B cells in 7 out of the 8 animals. And because these non-responders were different animals, if you combine the data sets, we see BG18 class B cells were primed in all 8 animals. And because we were just defining the BG18 class, as I said, based on that one sequence motif in the HCDR3 loop, so we wanted to validate that we were correct when we were saying that. And so Andrew Ward's lab at Scripps determined structures of 3 of the BG18 class antibodies that we elicited in complex with the immunogen. And so that's shown here. On the left is the HIV broadly neutralizing antibody BG18. Again, I'm showing that HCDR3 loop that reaches past the sugar molecules to contact the gray surface. And if you look at the 3 other antibodies that we elicited by the vaccine, they're basically identical. Their HCDR3 loops are doing exactly the same thing, reaching past the sugars and contacting the surface in the same way. So we were confident that when we call it a BG18 class B cell, that it really is a BG18 class. And in order to make broadly neutralizing antibodies, these are just precursors that we're targeting. We need them to affinity mature into BNAbs, as everyone so far has already discussed. And so were we getting somatic hypermutation? We looked at that over the course of 10 weeks after the immunization. We did get an increase in the BG18 class antibodies. By week 10, they're around 7% mutated in their variable gene. And so we think we're getting a good amount of somatic hypermutation. On the plot on the right, we looked at the binding affinity of the BG18 class antibodies to the antigen. And so this is an SPR plot, so which means lower on the plot means the antibody binds with higher affinity. And so going from each of these dots is one antibody. So going from week 3 to week 10, you gain about, it's like 4 orders of magnitude in affinity. So almost 10,000-fold increase in affinity for the BG18 class antibodies. So their affinity maturation is going very well. And just the last thing I'm going to say is that, so the next step, we need to boost them with a more native-like immunogen, as Bill and Facundo had talked about. And one feature of the vaccine is that it doesn't only elicit BG18 class antibodies, although that's what I've been talking about. It also elicits a whole bunch of other antibodies, which we believe have no chance of becoming broadly neutralizing antibodies. And so here we ordered like a random set of these, about 50 of them, and produced them, the ones shown in gray, and we measured their affinity for the immunogen N332GT5. And you can see they have similar affinities to the BG18s, which are shown in pink. But based on this data, we were able to develop a series of boost candidates. I'm showing one here, which are more native-like than the N332GT5 immunogen, so they're closer to a wild-type trimer. And they completely knock out binding to all of the off-target antibodies. Everyone that we tested does not bind to the boost candidates, although still more than half of the BG18 class antibodies bind. So we think we can selectively boost only the BG18 B cells. And so for conclusion, we validated that this we can prime the BG18 class antibodies in non-human primates with N332GT5 and SMNP. We think this is a good proof of principle for targeting HCDR3 dependent antibodies. And so based on this, we think this should work for essentially any antibody now, or not any antibody, but most pathogens will have antibodies that are good targets for germline targeting. And so the next steps are to develop a sequential boosting regimen so that we can now walk these BG18 class antibodies that we've primed. We need to walk them along to become broadly neutralizing antibodies. And we're currently been working on that for quite a while in non-human primates. So we think we're getting close to achieving that. We're also evaluating, we need to evaluate to see if we can elicit BG18 class antibodies in humans. And so that clinical trial is also happening. HVTN 144 is evaluating N332GT5 with SMNP, basically exactly what I described in non-human primates we're doing in humans. That's happening right now, both an escalating dose that I just talked about and a bolus immunization. And so I just want to thank everybody that worked on this project. There's many people from Bill's lab who worked on it. It was a big collaboration with Shane Crotty's lab and Devin Salk's lab. Andrew Ward's lab has solved so many structures for us. We've been working with Facundo for many years on this project, eliciting BG18 class antibodies. And the CHOB-D is the primary source of funding for this project, but also the Bill and Melinda Gates Foundation, Moderna. Yeah. So thank you, everybody.
Thank you. And our final presenter in this panel is Dr. Torben Schiffner. Dr. Schiffner, I think you're muted.
My apologies, and thank you very much for the invitation here. So I'm presenting on another antibody that we're following up on that's targeting the membrane proximal external region of HIV. So in this little cartoon here, we have the envelope protein in blue, and there is a particular region of it that is highly conserved. It's called the membrane proximal external region. The membrane is here down at the bottom, and the emperor is this pink region right next to the membrane. And this region is very conserved, but and therefore antibodies that target this region are usually very, very broadly neutralizing. So this antibody 10E8, which is kind of our prototype, our target, it neutralized 98% of all strains that it was tested against. So if we could induce it by vaccination, that would really give a nice broad protection. However, this AMPA region is so hidden away at the base of the trimer, the trimer actually has to kind of tilt over for the antibody to get in there. So it's really difficult to access this region. And 10E8 therefore needs a very long HCR3 loop to kind of grab into this epitope. And it's a specific binding motif at the tip of this long HCR3 loop to make contact with the AMPA. And that makes it really challenging to induce antibodies to this region by vaccination. So we took a strategy that is called epitope scaffolding to kind of take this little region out of its normal native context and make a scaffolding protein that stabilizes this region in the antibody-bound conformation. And now the scaffold protein, this is going to be much easier for the antibodies to get access. So it's easier for the immune system to see if we vaccinate with this, and therefore much easier to induce antibodies to this region. However, epitope scaffolding on its own is not going to be sufficient, because now we have binding to the mature BNAP 10E8 if you do this epitope grafting strategy. And again, similar to what John showed, good binding is at the bottom of these plots, no bindings at the top. But we still don't get binding to the precursors, the unmutated precursors of these antibodies. So we need to combine this epitope scaffolding strategy with the German targeting strategy that the other presenters have talked about before. And after many, many rounds of optimization, we have these two immunogens that we call GT10 and GT12 that not only bind the mature broadly neutralizing antibody 10E8, but they also bind to about half of the precursors, of the diverse precursors that have the genetic capability of maturing towards broadly neutralizing antibodies. And now we assemble this protein into nanoparticles, and we do all kinds of other engineering, and end up with an immunogen candidate that we'd like to test. But since it's targeting human B-cell receptors, it's not that straightforward to do the testing. So we paired up with a Quaddy lab, and what they did was, instead of just directly putting the vaccine into a human, they took B cells out of the human, and then in the lab in vitro, we can probe these B cells for the ones that bind our immunogen. And then they sequence the B cell receptors of the B cells that bind our immunogen and look, are these B cells 10E8-like? And when they did that, well, the first thing they looked at is how many have this 10E8 binding motif? And normally in this motif is found in about 2% of all B cells, but the B cells that bind our immunogen, it's over 80% of the B cells that bind our immunogen have the binding motif, so massive enrichment of the target B cells. The other thing is, I said we need this long HCR3 to reach into this kind of occluded region at the base of the trimer. And what you see here is the normal length distribution of B cell receptors, and specifically the HCR3, this long loop. And normal lengths are like 15 amino acids, but 10E8 needs a length of roughly 22. We give it a little wiggle room from 21 to 24, shown in gray here, but it needs to be really long. But not now, what about the B cells that engage our immunogen? Well, those B cells are highly enriched for these long HCR3 loops, pretty much centered right around the length that we're looking for, 21 to 24 amino acids. And so we get both the long HCR3s and we get the 10E8 binding motif. So we're really enriching exactly for the characteristics we're looking for. And the next thing we did was we collaborated with a number of labs, Facundo's lab and Ling Qian's lab, and we did mouse immunizations with our immunogen. And this was another humanized mouse model that can make 10E8-like precursors, and we immunize these mice and then look at what kinds of B cells are triggered by our immunogen. And first of all, if you again look at the length distribution of the B cells triggered in these genetically engineered mice, by our immunogens, you again see this, these long HCR3s that our immunogen triggers in these mice. And overall, about 5% of all the B cells that were triggered by our immunogen have these characteristics of 10E8 that we're looking for. And importantly, this worked both when we gave the protein, the immunogen as a protein, classical immunization adjuvanted protein, but we also teamed up with Moderna, and they made an mRNA LNP version of this immunogen shown on the right here. And the mRNA did exactly pretty much equivalent to the protein immunization in triggering 10E8 class B cells in this mouse model. So we think that this data show that we can prime B cells using these epitope scaffold nanoparticles that we engineered. But there's still a long way to go to get all the way to BNAP. Similar to the other speakers before me, we need to not only prime these rare B cell precursors that have the genetic components required, but we also need to walk them along and boost them towards broadly neutralizing antibodies. So we have a boosting candidate, a first boost candidate, and we immunize these same mice with these boosting candidates. And on the left, you see what happens if you just prime and don't give a boosting shot. You get very few B cells, 10E8 class B cells that are now capable to bind the boosting candidate. If you don't prime and just boost, you get virtually no 10E8 class B cells at all. Only this one mouse had a couple of them. But if you first give the prime and then you give the boost, then you get 10E8 class B cells in almost all animals. And we also did a pilot study in non-human primates, and similar to what John presented before me, primates are a really challenging model. B cells, 10E8 class B cells are more rare in monkeys than in humans. So it's really challenging. And nevertheless, we went ahead and immunized them with the priming immunogen first, and then we boosted with the boosting immunogen. And in all animals, we found 10E8 class B cells after the boosting. And the B cells increased their affinity upon boosting. So we kind of walking the B cells along on this path towards broad neutralization. So we kind of have a pretty good candidate now as well for this first boosting candidate, driving the B cells along on this path towards broadly neutralizing antibodies. And to sum all this up, I've shown you that we can make these kind of artificial epitope scaffolds that are engineered to specifically engage diverse precursors with our predefined features, the long H3s and the specific binding motif. I've shown you that you can use our immunogens to pull out B cells with these properties from human blood, and we can immunize both stringent mouse models and non-human primates and activate these B cells. And this works as both protein and mRNA. And we also now have a first boosting candidate that looks very promising. And therefore, we believe that these germline targeting epitope scaffolds are ready for testing in clinical trials. And with that, I'd like to thank everybody involved, same as the other speakers. This was a huge collaboration with many different labs involved that all did essential parts to move this project along. And of course, I'd like to thank the funders, without whom none of this would have been possible. And thank you for your attention.
So I want to thank all the members of this panel. Clearly, a lot of hard work went into multiple stages of evaluation and development to this stage, but it sounds like we're making progress. Our first question is from Dan Hoft.
First, I want to thank all of you for all this important work and for updating us on what you've been doing. It's all very exciting progress. I have two questions. The first one is, what reassurance do we have if we are successful in inducing broadly neutralizing antibodies to high titer in people that doesn't cause selective pressure on these epitope sites to escape? My second one is, are we giving up on T cells? Or what about CD8 T cells from internal conserved proteins as an additional strategy?
I
Please.
think that those are two very important questions. I think that the notion indeed of having multiple epitopes that they are going to neutralize the virus is the way forward, because even if the virus managed to escape to one, then you have the redundancy of the others. Indeed, when you look at people who are infected with HIV and they are treated with 3 different monoclonals, that reduce the amount of load. And these are people that are infected. So the notion will be that by inducing polyclonal, and that is what we are doing, Torben is working on one, John is working on that, Chris is working, by combining all of this in a single vaccine, that will make the possibility that the virus will have little opportunity to escape. And the second issue, at least from my point of view, regarding T-cell vaccines, of course, that is something that is becoming very popular these days. And the dream will be to have them both combined together. So you don't have only this polyclonal neutralizing, broadly neutralizing sera, but also you have the reassurance of T cells and also eventually NK cells working there.
James, please.
Yes, thanks, Facundo. I completely agree. And additionally, Dan, some of the work being done with the monoclonal antibodies, and there was the antibody-mediated prevention study that provided us a real target for what those in vivo levels of BNAbs is that we can target for future combination BNAb monoclonal studies, but also, what really threshold is critical for active immunization for these combination BNAbs that Facundo was talking about, using multiple antigens and epitopes. And then additionally, within the HVTN, we have now almost completed what's called HVTN 142, which is focused on evaluating the safety and immunogenicity of the CMV vector, which has a real, very specific MHCE restricted presumptive responses for CD8 T cells. And of course, the intention would be then to combine such a vector with the BNAb inducing antigens that you've heard about today.
Thank you.
If may I allow to add also, I was about to put this in the talk, but I didn't want to make it together again with the laboratory of Bill. What we show now is that this priming can occur together. So we have very strong evidence, at least in the mouse, that we can prime MPER, CD4 binding site, N332, and APEX all in the same vaccine at the same time. So the beginning is starting not only that we will regard multiple, but all this can happen already, and we are about to submit that paper again. There is a large collaboration with the Bill Lab.
Well, we don't have any other questions or comments in the room or online. I want to thank all of you for your presentations. It's a very complex field, but just amazed at the progress you've made, and look forward to hearing as we continue to move forward. Thank you for your time.
Thank you for having us.
Thank you very much.
Thank you.
So we'll now move to our discussion around the National Vaccine Strategy Charge working session we initiated yesterday. Anne is going to lead us in this discussion. And so Anne, you have the floor.
Thank you. So just as a quick reminder, our goals for this meeting were to sort of discuss the goals of the original, the previous National Vaccines National Strategy, which we did yesterday to some extent. I'll give you a chance if anyone has any other comments they'd like to make about the goals. Next, we'll go into indicators, including the 3 developmental indicators. And then one of the last things, which we may not have time for, and we can do this over email too, through some sort of fun voting, if you will, is to recommend and prioritize the objectives. And Emerald Levine had asked for 3 for each goal. that we think are poised to make the greatest impact on the U.S. immunization system over the next 5 years. And so here are the current goals. We had a very good discussion yesterday about trying to keep the 5 goals, but maybe doing some tweaking of the language, potentially also maybe reordering the goals. Did anybody else have any comments they wanted to make about goals before we move on? Go ahead.
Sorry, yesterday I hadn't done my homework and wasn't quick enough to weigh in. But I wanted to, I agreed certainly with safety being number one. Safety being number one, that's obviously critical. But I was wondering about what I was thinking was that moving the innovation aim way down the line really kind of de-emphasized that important area. So I was going to lobby for thinking about that as the second goal, but expanding it more broadly with language that would say something like foster innovation in vaccine development, implementation, and equitable uptake as an alternative statement or goal.
Any thoughts about goal 1 language, or what's currently goal 1 language? Okay, so we'll include that and we can, go ahead.
Andrew, please.
So I'd have to think about that language a little bit more, but I think that edit, would monoclonal antibodies as discussed yesterday still fall within that category, or would we still need to retain a language about related technologies?
At least in my mind, I think we have to keep monoclonal antibodies as part of our discussion and focus. You know, Ann and I have had this discussion many times over the last few years. You know, when I think about our immunization landscape, I almost think about a 3-legged stool. You know, one being our traditional anti-infective vaccine processes, one being monoclonal and other passive antibody products, and the third being potentially therapeutic vaccines. And I think all of those need to be, at least conceptually, within our model, or we're missing some real opportunities going forward for our society.
Okay, thank you. So here are the current objectives for each of the goals. And like I said, there's only 2 for the current goal 1. But support the goal, objective 1.1 is to support the development of innovative, safe, and effective vaccines to prevent infectious diseases of public health significance. And objective 1.2 is to support the development and uptake of technologies to improve vaccine manufacturing, storage, distribution, and delivery mechanisms. And so we've had a lot of INVAC sessions on both of those, but I just wondered if people had thoughts about these objectives. And some of the discussion questions are similar to what we talked about yesterday. Do we recommend keeping these with edits? Do we want to add edits? Are there ones you don't want to keep? Are there ones you want to add, et cetera? So,
Molly, how?
Yeah, for objective 1.1, I was just wondering, like the last NVAC meeting, we had a number of sessions on cancer vaccines. And so I was wondering if we needed to remove infectious or add some other language so it's not just infectious.
Yeah, this is one that directly related to your comment as well as Andrew's, where I've debated in my mind, do we remove infectious from 1.1, or do we have another objective that focuses on therapeutic vaccines, and potentially an additional objective that focuses on passive immunization products, such as monoclonal antibodies? And my inclination would be that those are each separate objectives. They're interrelated, but they're each have somewhat different implications. Dan, please.
I was underwhelmed by the response to my first recommendation. But just to add to what I was thinking, if we expanded this to something like a vaccine development, implementation, and equitable uptake, we should add a third objective that focused on the equitable uptake, which is not here.
And in our innovation report that we're working on and through some of the other meetings, funding has come up a lot. So I don't know, I'm just throwing it out there as a potential idea, but if we want to think about like creating a space where innovation's really easier or better or whatever. So I wanted to see if people had thoughts about that.
Foster is a very broad word. You know, does foster mean intellectually? Does it mean financially? Does it mean, I think there are all kinds of different pieces there. And it may come down to how granular we want to term things. Other thoughts? Mitch Rotholz online.
Yeah, I was just trying to type some more things, but I'll say verbally. So on the
Mitch, can you speak up a bit? We're having a hard time hearing you.
okay, can you hear me now?
That's better. Thanks.
Okay. So I put a comment about objective 2. Can we add maybe usage as well, not just delivery, but usage, using technology that helps with usage, but getting to the point about payment. Could we can't lobby, but can we facilitate resources or something from that perspective that supports the technology, development?
Good thought.
Anything else for what we're currently calling goal 1? Okay. So here's goal 2. We have 3 objectives. 2.1 is to minimize preventable vaccine-related adverse events. Objective 2.2 is to improve the timely detection and assessment of vaccine safety signals to inform public health policy and clinical practice. And objective 2.3 is to increase awareness, understanding, and usability of the vaccine safety system for providers, policymakers, and the public. So thoughts about this? And the same questions as for the other goal.
Dan, please.
Like I said before, I do think safety deserves to be goal 1. But I would consider language maintained to me as kind of weak. And so I would think about promote vaccine safety as the highest priority. Maybe it needs to be longer than that, but something like that as the first goal.
I think we've got a robust vaccine safety system, here in this country. My appreciation of that is at a very different level now than it was prior to the last 5 years, going through the pandemic and looking at our COVID-19 vaccine safety record. The only concern I have with us going toward a word that just focuses future, making safety bigger, is that I don't want us to minimize the value and the strengths of what we have at present. We've heard a number of panels and presentations through this committee over the last several years about different ways that we might leverage what we're doing now in the vaccine safety system, other international efforts around that. But I don't know that I've heard anything that is necessarily going to say we need to replace or redo what we're doing at present. I think certainly there are opportunities for enhancement. But I don't want to undervalue what we're doing at present as we think about.
I get that, and
yeah.
I was just thinking as you were talking that maybe maintain and enhance would be a stronger statement, and making safety priority number one or something.
Yeah, I agree that I don't particularly like the word maintain, but I haven't come up with a better verb to replace it that emphasizes the strengths of what we're doing now and the fact that we need to continually think about continuous quality improvement with everything that we do that's important.
Uphold.
Yeah, I always think whatever we do, we're going to need to use two verbs. If we're going to talk about current and future, I think we're going to need to, and whether we do uphold or maintain as the current state, but I think we also need to include something that is a forward-thinking verb as well. Karen, please.
Just to say that I completely agree with you. I was also made aware of the vaccine safety system and how complex and very important it is. And when I look at these sub-objectives, I'm also thinking, how are you going to measure some of this? Because that's what Dr. Levine is calling for. And I mean, we minimize preventable vaccines as much as we can, and it's hard to measure how you can do that better. So some of this might need to be reworded a little bit. I don't know.
And we're getting into the indicators, which are more measurable.
Yeah.
So.
No, personally, I think number 3 is wonderful. I would think a little bit more about number 1 and 2. Yeah.
As I look at number 2 in particular, I mean, number 1 is certainly one of our aspirational goals. It's the whole focus of our safety system, is to minimize any preventable vaccine-related adverse events. But as I think about number 2, I wonder if that may be, number 2 may be our opportunity for something that's measurable. At X time point or at Y number of doses administered, we get a formal readout from the systems we have in place of what are the safety signals or what are the concerns going forward. That may be something that's going to be a measurable that might be worth consideration.
Any last comments here for goal 2? Okay, I'm gonna keep it moving. So goal 3, this is to increase knowledge of and confidence in routinely recommended vaccines. Objective 3.1 is to counter vaccine myths and disinformation and increase public support for the individual and societal benefits of vaccination. Objective 3.2 is to increase provider capacity to promote knowledge of the benefits of immunization and increase vaccine acceptance by the public. Objective 3.3 is to ensure key decision and policymakers receive accurate and timely information on vaccines and strategies to promote vaccine uptake. And objective 3.4 is to reduce disparities and inequities in vaccine confidence and acceptance. Same questions for this.
So I might pose for goal 3, just something to think about. And again, my wording may not be ideal. Communicate the value, efficacy, and safety of vaccines in order to increase confidence in vaccination to benefit our society. Rebecca?
Yesterday, Mitch and I were sitting next to each other and talking a little bit about the role of communication and not necessarily seeing that elevated to this level. I think one thing that struck me as I was looking through this objective and specifically the objectives 1 through 4 is that there needs to be, I think there needs to be something that calls out hearing from the general public and how that is solicited, recognizing that communication needs to go both directions. Now, I don't know what the wording is on that. I'm just suggesting that we maybe think about how to incorporate something like that.
I'll call that a friendly amendment. Yeah. Because obviously communication is of no value if it's one directional. Jewel?
Harkening back to Stephanie Friedhoff's presentation from yesterday, I wonder about modifying objective 4 to state that an objective to reduce structural factors responsible for disparities and inequities in the vaccine confidence system.
So clarify that'd be objective 3.4.
Mhm.
Okay. Steve Rendernecht.
Yeah, just looking at the wording of goal 3, a thought here, instead of how it's worded, just a few small word changes I think would make a large impact in changing this to, instead of increased knowledge, state improve knowledge of and increase confidence in routinely recommended vaccines. Just kind of a switch of some of those words, but puts emphasis on both improvement in our knowledge base the public currently has, as well as increasing confidence.
And in addition, looking at these objectives, we've got at least a potential for measurability in objective 3.2. And in reference to our panel yesterday, certainly it requires some work, but thinking about some data-driven answers to assess how much we're assessing and improving those structural barriers and inequities.
Okay, thank you. Any last comments for the objectives in goal 3?
And
Oh, Mitch, go ahead.
yeah, I was trying to type, but I can't do type quick enough to get. If our goal is to get to 3 objectives, could we potentially look at taking objective number 1 and doing some tweaks to it that take objective 4 and include it in there, like something like address barriers and factors that contribute to in something about vaccine confidence or acceptance. So just basically try to that would also include addressing disparities and inequities. So I'm just trying to, if we're trying to get to 3 objectives, that might be a place to look for consolidation.
And just to be clear, they can have more objectives under each goal, but we're just supposed to prioritize the top 3 that we think. So you could make a recommendation around, we think there also needs to be this, or we agree with these other ones, but these are the top 3 that we've chosen.
Okay.
Jill, please.
Just in response to that idea, I think we would lose the essence of both or dilute them by lumping them. So just not in agreement.
And Chris Arisman online, please.
Actually, I must have accidentally bumped my hand. I'm sorry.
All right, you're off the hook.
And I'm going to try and move on to goal 3, but I actually can't, Rebecca. The clicker's not working any longer. Can you move on to goal 4 for me? And this one is the one with all the objectives in it. So the goal is to increase access to and use of routinely recommended vaccines as currently written. Objective 4.1 is to increase the availability of vaccines in a variety of settings. Objective 4.2 is to reduce disparities and inequities in access to and use of routinely recommended vaccines across the lifespan. Objective 4.3 is to strengthen data infrastructure, including immunization information systems, to track vaccine coverage in the United States and conduct surveillance of vaccine preventable diseases. Objective 4.4 is to reduce financial and system barriers for providers to facilitate delivery of routinely recommended vaccines. Objective 4.5 is to reduce financial and system barriers for the public to facilitate access to routinely recommended vaccines. And objective 4.6 is to promote public-private partnerships to increase the capacity of the health system to deliver vaccines for routine use and protection during outbreaks. So there's a lot here. The thoughts?
You know, in the context of some of our earlier discussions, and looking at this list of objectives along with the goal, I almost wonder if we don't need to have a goal focused on financial and systems barriers, elimination of financial and systems barriers within the public immunization space, public and private immunization space, and the other focused on the actual immunization piece. You know, you almost need to think about barriers separate from immunization. See a few nods of heads.
You could combine 4 and 5, which is delivery and access, no?
Yeah, it's all financial and systems barriers, but it affects multiple constituencies. Rebecca, please.
I had one comment related to, I think, objective 4.3 and strengthen the data infrastructure. I don't disagree with anything that's stated here, but I am also recognizing that what's perhaps missing is the access by providers to this information to make better decisions in conjunction with the patient, and also patient access to their own records.
Mitch, please.
Yeah, so two points. Just one that on following up what Rebecca just said. I think it's strengthen and increase usage of data infrastructure. I agree with what she said. It's not just that it's there, but it's usage by providers and the public. And then the other part going to the finance 4.4, I think we should make some kind of statement or inclusion in there about something about sustainability of community-based providers. And I think, Bob, that gets to your point about the impact on system from the financial support and all that. I think what we're in jeopardy if we don't put some attention to it is losing those community-based providers, whether they be medical practices or other clinics or pharmacies in the community. If we don't address that as a nation, we're not going to have it available for when we need it.
Good point. Molly?
Just adding on to what Rebecca said, I'm wondering if under goal 3 that we were just discussing, if you could add something about IAS there and their use to support providers and the public in the, like more of the clinical setting, and then you could potentially leave the IAS objective 4.3 here to talk more about using it to track coverage and uptake.
Mitch, do you have another question, or is your hand a legacy hand?
Legacy.
Gotcha.
Okay, so I'm going to move us on here to goal 5. Protect the health of the nation by supporting global immunization efforts. And objective 5.1 is to support vaccine research and development to address vaccine preventable diseases of global public health importance. Objective 5.2 is support global partners in efforts to combat vaccine misinformation, disinformation, and hesitancy worldwide. Objective 5.3, support global partners to strengthen immunization systems. And objective 5.4 is to increase coordination of global immunization efforts across federal agencies and with global partners. So thoughts here?
Yes, we at USAID were super happy to see goal 5 represented here, and I'm glad that yesterday we confirmed that we want to keep it. A couple of thoughts from us instead of hesitancy to use confidence and to continue that language throughout. And then as well as maybe further defining support in a couple of these objectives. They're pretty broad. And sorry, last one is on global partners in 5.2 to ensure that also means partner countries, not just WHO and UNICEF, but also the governments that we need to be strong partners with. Thanks.
And again, here's an opportunity for us to think about something that may be measurable is, are we engaging countries, non-governmental organizations, the public, all of which are going to be important. Yeah. Molly, please.
Yeah, and I don't know if it would be best here on the safety one, but especially during the COVID pandemic and vaccine rollout, the use of looking at other countries' vaccine safety surveillance systems and how powerful a lot of those systems are, it might be an opportunity to kind of lift up some of that collaboration with other countries on vaccine safety surveillance.
Clearly, that was a critical piece in our COVID vaccine safety assessment was interactions with our global partners in safety. And at least my opinion would be I'd put that in the safety mode, but having to recognize it is not just us. We're part of a global community in all of these aspects.
Other comments?
Jill, please.
Not necessarily a suggestion, but a recollection that during the planning for the vaccine innovation report, we spent a lot of time talking about global equity and what that meant. So without adding an objective, I thought there could be a place here to at least insert the recognition that part of this objective, the goal is really around equity as well. And maybe we can attach that to one of the objectives.
Do you see that as part of like objective 5.3? So support global partners to strengthen immunization systems and equity.
Yes.
We agree with that. Seeing equity as a real strong component for 5.3, I think, and adding the word would be great.
Karen, please.
I don't know if we're done with this one because I'm going to jump back to innovation. Maybe I don't know where I put this, but one of the gaps that we're seeing is special populations, and we talked a lot about maternal immunization, but also immunocompromised patients, and how we don't have enough data for certain populations and use of vaccines that are even available for the rest of the country. So maybe it will be an opportunity to highlight that as well.
And the heterogeneity of the immune compromised population and their immune responses. Absolutely.
I think that's a big gap. Yeah.
Okay, thank you. I'm going to take us to just a real brief, we've got about 3 more minutes to look at indicators. And like I said yesterday, there are 5 pediatric indicators here. They are tied to data sources to be measurable. There are targets for 2025 and 2030 and some baseline data as well. And I'm just going to let you guys read these. I'm not going to read them out just in the interest of time, but let me know when you're done reading. And maybe we'll just cover pediatric now. So any thoughts about these indicators?
So these indicators are useful. They're data that's reasonably easy to obtain. I would hope that in our current era, we might be closer to, if we're thinking about children who are up to date on a certain vaccine or not, that we might be able to use registry data rather than having to rely on survey data. And I might be more focused on some of the declines that we've seen in things like measles, mumps, rubella vaccination and the resurgence of disease, and trying to think about where we're needing to, I don't know, I need to formulate my thoughts a bit, but I'm concerned more about declines in rates that are having adverse effects on the health of our population and how we might measure that maybe a little more effectively. Rebecca, please.
I would potentially suggest adding an adolescent measure in there as well. So that very top measure or indicator is for children age less than 6 years of age. There's also, using the same data source, the availability to collect that for the adolescent population as well, which is IIS data.
And potentially what we measure is the things we're doing worst at, so that we really are focusing on improving where we have the biggest gaps. So potentially HPV for adolescents. We think about MMR for young children. And we've got multiple different examples of what we could aim at for adults. Molly, please.
Yeah, and I was going to get Rebecca's feedback on this. I was wondering, since we have good number of children in IAS now, even if it should be assessing the number of immunizations they have, like how many children, like 2 or more immunizations. So you're more assessing the data completeness, and that's also on the ISAR that we complete every year. And that would be my same comment on the adult measure also, probably.
Which, just to, I think solidify your comment there. So for the pediatric measure and the adolescent, you have to have 2 or more doses in an IIS, whereas adult is 1 or more, and they have to be specific to that age cohort. So you have to have 2 or more pediatric, 2 or more adolescent, and 1 or more adult vaccines.
Okay, so Rebecca, do you see our public commenter on the line? Okay, so we are going to stop the discussion for now. Thank you very much. I did want to say that you should not spell, or at least I should not spell on the fly. Can you advance my slide to the last one? So I guess I misspelled syndemic somehow yesterday. My apologies. Again, should not spell on the fly. So this is the email address. I send it out to all the members, but for anybody watching online or in the room, if you would like to make a comment about the vaccines national strategy or ask to be a part of one of the listening sessions that we're having, it is syndemics, SYNDEMICS at HHS.gov. And I will correct the record for that. So please feel free to use that if you'd like to. And with that, we will go to our public commenter, or I'll turn it over to Bob for that.
All right. Rebecca said we do have our public comment person on the line. Okay. The phone. So
please press *6 to unmute. Timothy Cicero. Are you on the line?
Yes. Can you guys hear me?
We can. Thank you very much. Please go ahead.
All right. Thank you. Good afternoon, everyone. Sorry, I was a little rushed yesterday. I was trying to set up for a kid's soccer practice while trying to talk at the same time. Not a good combo. So a little background. I spent 20 years in the Navy, and I was actually assigned to the DHA as part of the COVID task force and making sure that vaccine was distributed to military family members on time. And unfortunately, while I was trying to serve my country, my youngest son, Joseph, the youngest of 4 boys, he was at home struggling with some pretty severe health issues going on. And the doctors didn't understand really well, they did, but they downplayed what he had going on to routine eczema. Like if you and I stepped out of the shower on a cold day and a couple of days later we have a little spot of eczema, that's what Joseph was being categorized with. But the reality of what Joseph was suffering from was he had a poor functioning immune system. And I'm so thankful that you guys, in your objectives are now talking about safety, because that is what erodes the trust of people. No different than society getting eroded from maybe police officers not practicing the law properly. But pediatricians are not, and another thing that was just talked about, objective 2.1, you guys are talking about maybe how you can prevent some of these adverse reactions when they're preventable. Obviously, there's times where they're not, but we just touched on it. We need to bridge the gap of when immunocompromised infants are getting live virus vaccines. There is way too much discretion, way too much leeway, and doctors are not erring on the side of caution when it comes to these live virus vaccines. And straight from the CDC, severe complications have followed vaccination with certain live attenuated viral and live attenuated bacterial vaccines among persons with altered immunocompetence. Persons with most forms of altered immunocompetence should not receive live virus vaccines, MMR, varicella, MMRV. And it goes on. So how does my son that has, what does the CDC list as signs of a primary immune deficiency? They list recurrent infections, recurrent skin infections, recurrent ear infections. So does the Mayo Clinic, so does the Boston Children's Hospital. So how does my son that had 7 recurrent infections of the skin that were horrendous, that required multiple types of antibiotics and creams to heal, and had 3 recurrent ear infections leading up to the MMRV, my wife goes in that doctor's office. We think it's a well visit to see how we can get our kid better. How does that kid get issued the live virus vaccine coming off amoxicillin 2 days prior? That's how you erode the trust in the public, because my son now has brain damage from it. All my other kids were vaccinated with no issues, but when we're vaccinating kids that have an immunocompromised system, and we could live a lifetime and try and figure out what primary immune deficiency my son had, but we do know that he had an immune deficiency because his skin and his viral load in his body told us that. So if I figure this out as a police officer, there's 30 people in that room. The CDC language says he shouldn't have got that live virus vaccine. But why did he? Because doctors are being put under the gun in some way, sense, or form to vaccinate according to a time schedule that doesn't give them a lot of leeway to maybe make their incentives that some of the healthcare providers talked about yesterday. There's incentives for them, but maybe I didn't become a commander in the Navy to not just lay a whole bunch of problems on you. Here's the solutions. If a kid is showing with recurrent signs of infections, like my son did, maybe we tell doctors to send him to an immunologist and run the panels, the blood panels that the CDC recommends. So I don't know how that didn't happen, but it didn't. That would be one solution. If a kid has signs and symptoms of a primary immune deficiency, hey, all pediatricians in the world, do us a favor so we have one less brain damaged kid out there, send them to an immunologist. And 2, another solution, all these healthcare companies talking about how they're making sure providers are compensated. Why don't we spread out the childhood vaccine schedule to maybe 2.5 years, 3 years, give them a little leeway. So a kid that is presenting with signs, we could take a little time to discover what he's undergoing before he gets the live rotavirus vaccine that devastates his gut. And now he has a whole bunch of other issues, like his T-cell response is going to be mediated and a whole other host of issues that come with an infant with a poor, awful gut microbiome because you gave him 3 live rotavirus vaccines in a row. So this is just the research that I've done in the last 6 months since I discovered what happened to my son 3 years ago. It's all stuff you guys, I'm sure, understand and digest, but I'm offering solutions. The verbiage that is getting out to pediatricians is not sufficient. The CDC laid out specific requirements, but they are interpreting it in a totally wrong and inappropriate way, where they're not erring on the side of caution. They're erring on the side of I need to make sure this kid gets fit in the vaccine schedule before I don't get paid out on that incentive that is provided for me. So these are the problems that really face the Vaccine Advisory Commission. You want to erode trust? Have a kid like Joseph Sestaro, a beautiful little boy that is now has an altered path in his life, when that shouldn't be the case. And now, affect an entire family trying to figure out how to help this little boy. And there's ways to do it, and I'm trying. And, if we really want to help Joseph, if somebody could please email me or send me a comment, I would love to be part of the discussions that help you guys make this planet a safer place. But putting doctors in that time frame to get kids vaccinated is a tough thing for everybody. And it doesn't allow for a kid like Joseph, who is showing all these signs and symptoms, to get properly treated as an individual infant, not just as a mass thing. And, please, I need assistance with this little guy. I need assistance, feeling good as a dad that I didn't serve my entire life in the Navy, 20 years and 20 years as a police officer, to not protect my son and then not protect the next kid that comes after him. That the story's been heard a number of times. My son's just the latest, and I feel a little foolish that it happened to us. I hope you guys have my email. Please take my number. Please reach out. But this happened, and there's solutions to it. There really is, and it's figuring out how to get the CDC language down to the pediatricians. I talked to multiple pediatricians in the last few days, and all of them say, we're vaccinating sick kids. That's what one pediatrician told me on the phone yesterday. He has 3 practices under him. We're vaccinating kids with the live rotavirus vaccine that are sick. I was like, wait a second. That's not what the CDC writes. They write kids that are moderately ill and severely ill should not receive live virus vaccines, including that one. So why did my son, who had an open wound, allergic reaction all over his body that they were using mupirocin for, and cortisone and a bunch of other creams, and a puffy swollen face, get the live rotavirus vaccine? I know I'm going past my time. I appreciate you guys taking the time to hear my story. I hope somebody from this committee will reach out to me in regards to what took place.
Thank you, Mr. Sestaro. We appreciate your comment. Well,
and I'm glad I had a profound effect that you guys are now talking about safety as your number one objective. Good to hear that back on.
Thank you. We have to move on. Thank you, sir. We have an additional public comment.
No, I sent out a written public comment to everyone. You should have that in your email.
Well, this brings our meeting of the National Vaccine Advisory Committee for September 2024 to an end. I want to thank the participants that were online, as well as those that are here in the room. As I've said earlier, it's been my absolute pleasure to chair this committee. I know that with Ann and the folks here at HHS and the new chair coming forward, that you will continue to carry the torch and continue to enhance the vaccine system in the United States. Thank you again. This ends our meeting.
Produced by the U.S. Department of Health and Human Services.